Swayam Prabha. Digital India. Educated India.
Welcome back to the lecture series in animal physiology. So we are into the week 8. So we have finished 3 lectures, and if you recollect, in the last lecture we talked about the neuromuscular junction, the last segment of the stretch reflex arc. Or as independent also, there are so many neuromuscular junctions all over our body where the neurons are controlling the movement of the muscle, and there we kind of marry that part of the muscle where I taught you about how the sliding filament happens between actin and myosin filament, which is negotiated or executed by the calcium ions. And in the last lecture I told you from where these calcium ions are coming, because of the sensitiveness of the calcium ions via the ryanodine and dihydropyridine receptors. So today what we'll be doing is that we'll start talking about the special senses. So in the nervous system, this is the kind of the last tail piece of 3-4 more classes where we'll be dealing with special senses and sympathetic and parasympathetic system. Post that we'll move on to the cardiac system and then followed by the digestive system, like so on and so forth. Okay. So talking about the special senses. So just before I proceed further, this is our week 8 lecture 4. Okay, so we are starting with the special senses. Among the special senses, what we'll be dealing with are ear for hearing, then eyes for vision. We have talked about the sensory receptors like stretch reflex and all, which are handling your kind of a pressure or the stretch on the skin or in the muscle. Then we'll talk about gustatory receptor, that is very briefly about taste, and very little bit which human being does not use though, olfaction through the nose, olfaction or smelling. This is more of canines which are specialized on it. So these are more or less the 4 special senses we'll be dealing with. So talking about the modalities in terms of the hearing, this is pressure wave, a mechanical wave which is coming. Vision is an electromagnetic wave which is light. Pressure wave is sound. This is taste molecules. So they have a specific sites, and these are volatile molecules. These volatiles comes and bind to the odorant receptors which are present in the nose. Before going into the detailed anatomy and the physiology of this, let's talk about some of the very basic fundamentals. So the way it works is you have a sensor. In terms of all of them, which are the sensors? So you have the eyes, you have ear, you have tongue, and you have the nose. Okay, so these are the different sensors. These sensor apparatus has very specific sensing elements which upon interaction with the particular wave or particular molecule which have been listed out here generates a ionic current which activates the sensory neurons which are supposed to carry the signal. So this sensory input goes all the way through the spinal cord, all the way to the part of the brain where these informations are coded. So in the case of ear, it is auditory cortex in the brain, auditory cortex in brain. In the case of vision, it is visual cortex in the brain. In the case of taste, it is gustatory cortex in the brain, and for olfaction it is olfactory lobes which are present in the brain. And these areas, they are having the motor neurons which are sitting there. They process these different stimulus which are traveling from your eyes, ear, nose, tongue, skin, wherever, all this. And not only that, even from inside your body, as we'll talk about the gut physiology, we'll talk about places where there are acid sensing channels. If there is an increase in the acidity or something, those channels senses it. You have the pain sensing receptors, you have the baroreceptors. There are a series of such receptors which are there. But the architecture is similar. There is no difference in the architectural fashion. It's only the modular element changes. Okay, so as long as you keep this brain map intact, you will never do a mistake. So the sensory neurons carry the signal all the way to the brain, that respective area, right? What I just enumerated for you people, the auditory cortex for hearing, visual cortex for vision, gustatory cortex for the taste, olfactory lobe. There you are having this motor neurons which are sitting, they process the signal out here, and then they decode it and let you know what is happening. So it's pretty much the same thing as we talked about the reflex. Of course, the only difference is that between the reflex where the processing is exclusively taking place at the spinal cord level, here the processing is taking place all over into the brain. So now let's talk about the anatomy of this hearing, ear. So if you look at the ear is something like this, okay? This is the external ear. And I recommend you please go through any textbook, you'll see this picture much better than I can draw here. So this is outer ear. And the wave front travels like this along it. Okay. Now, you have the middle ear, so the membrane out here, and you have the middle ear where you are having three different bones. Something like this. Malleus, incus, stapes. These are very soft bones. Malleus, incus, stapes. So these pressure waves, which are essentially vibrations, these are now transmitted through these bones, which these bones constitute the middle ear. So outer ear, This is the middle ear. Now from the middle ear, the signal enters into cochlea, which is your inner ear. And the structure of the cochlea is something like this. It's something like during your childhood you must have seen, we have, when you go to this fair or something, you blow it like this and something opens out and it comes back. This is a curling thing, something like this. You must have seen in all these fairs, like, you know. You've seen this. So you blow air. Once you blow air, this stuff kind of, you know, becomes straightened like this, and then as soon as you remove the air, it again curls back to its original position. So exactly a very similar kind of, this kind of structure which is present in the cochlea. So our concern is the cochlea. Now if you look at the cochlea, the structure is very interesting. Underneath the cochlea out here, so this is kind of a very interesting structure. So you have these sensor elements. So these black spots are showing you the sensor elements which are present there. Let me blow up this picture, okay? Here you have the, I'll keep it, I will not make it very complex so that you can understand, but this is way more complex than this. Okay. So now, your sensor elements are sitting underneath like this. So the sound wave which is coming, the pressure wave, contraction and rarefaction, is traveling like this. Okay, so this is a semi-fluid filled cavity, and all your sensor elements are sitting here, and these sensor elements, they have a very peculiar shape. So let's detail out the shape of it before I'll come back to this picture. So sensor elements looks like this. Okay, and you see this hair-like structure. So these are called, so this sensor structure is called hair cells. These hair cells have this long processes. This is called stereocilia, and these other smaller processes are called kinocilia. Okay. Now underneath it, I'll come into the detail, don't worry. Okay. Underneath out here, you have something like this. Or if I make it slightly smaller, you will appreciate it better. I'm just drawing it at a smaller scale. So here you have the hair cells which are sitting here, shown by the arrow. Here you have the auditory nerves. These auditory nerves carries the signal all the way to the brain, and from the brain the decoded message, so the decoding is taking place at auditory cortex, and from there the signal is being decoded back, right? So what I wanted to tell you is this: in this circuit where all you could have problems, so one of the places, say for example your hair cells are, I haven't talked about the function, your hair cells are not functioning properly, so the no message will go. Or your hair cells are all fine, but your auditory nerves are not functioning, so that case also signal will not propagate to the brain. Okay, now think of a situation, this thing is fine, hair cells are fine, your auditory nerves are fine, but this auditory cortex is not functioning right, then too you won't be able to process the signal. But assuming that your auditory cortex is doing all perfectly fine out here, then you can bypass this whole route. This whole route you can bypass, and you can directly connect it to a mic, artificial mic. And if you have a way to integrate the auditory cortex with this mic, you will be able to listen. Not with that efficiency as the real hair cells, but you can, and that falls under the prosthesis. The reason why I, without explaining this, I came to this part explaining about hair cells, because ear prosthesis was the first success story, or among the very first success story of interfacing the brain with electronics. In the history, and most of these experiments or most of these successful stories had their origin somewhere in DARPA, which is Defense Advancement Research Program of US government. So some of these prostheses were developed there, functional one which really were implanted. And the reason to go for these kind of prostheses was very straightforward, because there are several soldiers in the field who get these kind of injuries because of sound of the shelling and all those kind of things. So they incur injuries either in the auditory cortex or in the hair cells. So how to bypass it? So they have these kind of prostheses there. So now, having said this, so you know the circuit, the way it works. So now, one take home message I will tell you first before I explain the hair cells, how the hair cells works. So these hair cells, each one of these individual hair cells which are arranged like this all along the pathway, and same all along this pathway, codes for frequency. The take home exercise for you to figure out, this happens in sequentially. So now figure out whether the higher frequencies are coded here in this part, which is the beginning, or the lower ones are coded here. Because if the lower ones are coded here, then eventually the inner will code the higher ones, or if the higher ones are coded here, then the lower ones will be coded inside. Figure that out. That I will request you figure that out. Now I'll tell you how these are being, how the mechanism works. So now each one of these hair cells which are present here, they are coding for, or a population of them are coding for a certain frequency. So let's assume that from the lower to higher or higher to lower, whichever way. Okay. So they are coding for nu 1, nu 2, nu 3. I'm representing the nu 4, nu 5, nu 6, nu 7. Either it could be ascending order or it could be descending order. Fine. Now these cells are very interesting. These cells, when they get, so for example here, when the wavefront moves, so you see there are bulges happening on this, like this, because the wavefront is moving. And this bulge will be coded by a certain frequency. And if the matching frequency comes, so this will lead to the bending of these hairs. And this bending is a very unique space phenomena, because if this one bends in one direction, say for example this bends like this, all of them will bend. Rest all the other kinocilia, if the stereocilia bends like this, then all the kinocilia will bend accordingly. And if the stereocilia bends on this direction, all the kinocilia will bend accordingly. And depending on the direction, whether they are bending on this direction or they are bending on this direction, either it will be on or off. On or off means if they bend in one direction, say for example, this will activate a series of mechanosensitive sodium channel. And if the mechanosensitive sodium channels are activated here, there is an influx of sodium and cations out here, it will generate an action potential, and that action potential from here will be transmitted to the next neuron, and this will be carried to the brain. And there will be a unique frequency coding which will happen because this particular hair cell is coding for a unique frequency. Now think of it, if you have a sound coming at a certain frequency, certain A, there are certain part of the system which will be responding to it, and there are other parts which will not respond to it. Or if you have a mixture, then it will distribute that which part is going to respond to which part. So this is how your hearing happens. So these are, so the take home message is very straightforward. Your sensor elements are hair cells. Hair cells have kinocilia and stereocilia. Stereocilia decides in which direction they will move. Depending on its directionality, whether it will be on or it will be off is being decided, right? And this generates a train of impulse. That impulse is carried from the hair cells to the auditory neurons, and from that auditory neurons it is taken to the brain, and the message are being coded, and accordingly we kind of realize what is the message all about. Okay, so please do the homework what I have given you. Just try to figure out that how the frequency coding happens on that circuit. Higher frequency to lower frequency or lower frequency to higher frequency. Please do that, okay? And apart from the function of the ear on this, there is another function of the ear. So this cochlea, what you see, has a one extension like this. This extension part, what you see, does not involve in hearing. This part is involved in balancing. It's something like, you must have seen there are people who experiences a vomiting feeling while going up the mountains, or kind of continuously looking up like this, you feel those feeling, you about to vomit and all. Such thing happens because here you have some kind of fluidic motions which are happening, and these are transmitted to the brain. So there is a balance area which is involved, and the crystals which are involved in it is called otoliths. There is a movement of these otoliths which regulate the mechanosensitive sodium channels to act, and based on that the balancing act happens. So ear is not, so you must have seen there are people who put cotton plugs while climbing mountains. Okay, so overall summary is that the basic circuit architecture remains the same, but it's just how it is clubbed together. Okay, so I'll close in here. Thanks for your patient listening.